Good morning, and welcome to the second of 3 videos on the neuron, which again is the beginning of chapter 2 in your textbook. Um, Now, today in this video, we're going to talk a little bit about what's called the myelin sheath. And you'll hopefully remember that I mentioned this earlier in the first video. But the myelin sheath is shown by these little white bumps here, okay, and here as well. All right. Now, on a sensory neuron, you find myelin wrapping around not only the axons but also the dendrites. You don't find myelin inside the brain or the spinal cord, but you do find it on the axons of motor neurons. So, what exactly is this stuff, and what is its purpose? Well, the myelin sheath is actually a layer of fats. All right, lipids are the fancy biological word for fat, and so it's a lipid covering on long axons that acts to increase the speed of nerve impulse conduction, insulation, and regeneration in the peripheral nervous system. All right, now um, the substance itself is produced by cells called Schwann cells, which are also sometimes called oligodendrocytes. I love that word. All right, say that 10 times fast. Oligodendrocytes. Oligodendrocytes. All right. In any event, these are types of glial cells, neuroglia, that actually produce the myelin sheath in the peripheral nervous system. Now, as I said, 3 major purposes: nervous impulse conduction, insulation, and regeneration. Now, think about, let's talk about the insulation part first. All right, when you have an electrical wire that runs, for example, from a lamp into the plug in the wall, all right, you don't want bare metal there for 2 reasons. First of all, it'd be very dangerous because you could electrocute yourself if you happen to touch that wire. But secondly, you also want to hold the electricity in the wire so that it gets from the plug to the lamp, right? You don't want it to be, going into the floor or the wall or, whatever that wire happens to be touching. And so we coat it, we insulate it with rubber or plastic. The same kind of thing happens in the neurons, and that's why we cover them with myelin sheaths. Now, you may have noticed in the picture on the previous slide, there were gaps between the sections of myelin, and these gaps are called the nodes of Ranvier, which is actually the name of the guy who first described them. And what they do, these gaps and the myelin together, what they do is to allow what we call staltatory conduction. And so what happens here is that the nervous impulse doesn't just start here and go from one end to the next of the neuron. What it does is it actually jumps from one node to the next. All right, and it just keeps jumping. Now let's go back to this picture. So the electrical impulse will actually jump from here to here, and from here to here, and from here to here. Now think about this. If you were walking across a room, if you had to go heel to toe and literally put, now these are not feet, obviously, so just imagine, here's one, you put the next foot here, and the next foot here, and the next foot here. It would take you forever to walk across the room. All right. Instead, you make big, long steps, and there's a gap, there's a space between your 2 footprints. All right. That allows you to walk much faster. Well, the same kind of thing is going on with the myelin sheath. Because we're doing staltatory conduction and jumping from one node of Ranvier to the next, all right, that allows the impulse to travel much, much more quickly down the length of the axon. Okay, and so not only does it insulate the axon, but it actually increases the speed of the impulse on an axon. Excuse me. Okay, so the nodes of Ranvier are the gaps between the myelin, and that allows saltatory conduction. Okay. Now, you may have heard the term white matter and gray matter when referring to the brain or the nervous system. So what exactly does that mean? Well, you may have noticed from the picture that the myelin is illustrated in white. And there's, that's no accident. All right. Myelinated axons are considered to be white matter, whereas unmyelinated axons are considered to be gray matter. All right. The gray matter is mostly the central nervous system, the brain and the spinal cord, whereas white matter is mostly found in the peripheral nervous system, which consists of the sensory and motor neurons, all right? So the sensory and motor neurons tend to be myelinated. The interneurons tend to not be myelinated. Now, why is this important? Well, again, we went through the main purposes of this, insulation and speed of conduction, right? But if that myelin starts to deteriorate, or has any other kinds of problems, we can end up with diseases. A prime example of this is the disease called multiple sclerosis. And this is a deterioration of the myelin sheath. And when the myelin deteriorates, it actually inhibits the impulse from traveling down the length of the axon. And so that gives folks with multiple sclerosis a really difficult time, especially with motor output, moving and controlling the muscles of the skeletal muscles on their body and allowing them to move. Okay, so this is another Mr. Anderson video from Bozeman Science. And so you can't actually view this through my video, but again, from the PowerPoint that you have on the in Canvas, please download that and then click on this link so that you can watch the Mr. Anderson video on neurons and nerve conduction. Okay, so we've talked now about the how chemical signals travel down the length of a neuron, from the dendrite to the cell body to the axon. What happens when they reach the end of the axon, and that signal then needs to be transmitted either to another axon or possibly to a muscle or a gland or some other organ in the body? All right. Well, the electrical signal cannot actually jump across the gap that exists between two neurons, for example. All right. And so when an electrical impulse reaches an axon terminal, it is translated into a chemical message, which is then released into the gap between the two nerves. Okay. So if you've got a neuron here and a neuron here, all right, the electrical signal can't jump from here to here. Okay. It has to release chemicals that then diffuse across the gap and eventually then stimulate a new electrical impulse in the next neuron. All right, and so the signal is constantly being changed every time you reach the end of a neuron, from electrical to chemical and then back to electrical. All right, because again, the electrical signals can't jump across that gap. Now, the gap itself is called a synapse, or sometimes called a synaptic gap. And it's defined as a small gap between the sending neuron, sometimes called the presynaptic membrane, and the receiving neuron, which is sometimes called the postsynaptic membrane. Pre for before, post for after. All right. And so you've got to get a message from the presynaptic neuron to the postsynaptic neuron. Okay. How do we do this? Well, transmission is accomplished across this gap by a chemical that we call a neurotransmitter. All right, and there are some examples of these listed here. Acetylcholine is the ACH, acetylcholine, dopamine, and serotonin. And there's a few others as well. We're going to talk about each type of neurotransmitter and exactly what they do in the third of these 3 videos. Now, the neurotransmitters are stored inside the nerve cell in special containers that we call synaptic vesicles. And essentially what happens is that when a nerve impulse reaches the axon terminal, a chemical change occurs in the plasma membrane, the cell membrane of the presynaptic nerve cell, and the neurotransmitters are released. And they diffuse across the gap, and then they reach the postsynaptic neuron, and then they either generate a signal by being excitatory, or they stop the signal by being inhibitory. Okay? Generally speaking, the neurotransmitters then return to the original synapse, to the gap, where they're either destroyed by enzymes, right, digested essentially, or taken back up by the sending neurons. So they can be recycled and used over and over again. All right. So again, the electrical signal reaches the end of the sending neuron. The sending neuron then empties its synaptic vesicles of neurotransmitters into the gap. Those neurotransmitters diffuse across the gap and either stimulate or inhibit a new electrical signal in the postsynaptic neuron. All right. And so again, the signal goes from electrical to chemical to electrical again. Okay. So this is an image of how that actually works. All right. The big lumpy thing here that kind of looks like an upside down mushroom is the sending neuron. All right. And if an action potential arrives at the end of a neuron, a couple of things happen. First, after an action potential arrives at an axon terminal, calcium enters, all right? And you can see the calcium ion shown here, and it's going into the axon terminal, the end of the axon. And what that does is it causes the synaptic vesicles, all right, there's one right here, to fuse with the synaptic membrane. And all the chemicals here are then released into the gap. All right, and so this space right here, that's the gap between the axon terminal of one neuron and the dendrite of the next neuron, for example. All right, so the neurotransmitter molecules are released, and eventually they bind to receptors here. All right, they bind to receptors on the postsynaptic neuron, the receiving neuron. All right, and so the little blue dots in here represent those neurotransmitters. Now, when an excitatory neurotransmitter binds to the receptor, so only for an excitatory one, that causes sodium to diffuse into the receiving neuron. So the orange dots here represent sodium, and the sodium will go in, and what that does is it changes the electrical composition, right? Because if all those positively charged sodium ions are outside the cell, and all of a sudden they go inside the cell, that reverses the polarity there, all right? And so the sodium diffuses into the receiving neuron, and a new action potential then begins, all right? If you need to, go back to the video on action potentials and watch that again so that you can see the relationship between the sodium and the potassium and how an action potential actually carries the signal down the length of the neuron. Okay. So that, my friends, is actually where we're going to leave it at that for video number 2. So please stay tuned for video number 3, and I hope you learned something.